Thank you everyone for joining us today and welcome to our webinar on navigating the advanced therapies for supply chain insights, practices and distinctions. So my name is Victoria Mwanza. I'm a global change facilitator at BioPhorum, and I'd like to thank you all for joining us today. Myself and our guest speakers today, Amanda, Donnie, Leslie, Tom, Ike. Today is the first in a series of webinars exploring supply chain cell and gene therapy, insights, practices and distinctions. In the webinars today, we will explore at a high level the regulatory landscape, choosing and defining raw materials, right the way through to finally completing supplier selection and qualifying a supplier. Today is the first in a series of 3 webinars. We have our webinar today, which gives a general overview, followed by a second webinar that delves more deeply into MRP and supplier relationship management, and then a third webinar which will look at some MRP tools that small startups, people that are new to the cell and gene therapy industry can use. So before we start the webinars today, I'd like to just tell you a little bit about BioPhorum. BioPhorum's mission is to create an environment where our members from the global pharmaceutical and device industry connect, collaborate and accelerate progress for the benefit of all. BioPhorum currently has 12 forums with over 150 companies as members. By coming together and collaborating, companies are able to accelerate change better, faster and cheaper than it would be for individual companies to do it on their own. Today, we are part of the Supply Resilience Forum. So the team today presenting is the Supply Resilience Forum, and we're part of the Cell and Gene Therapy Materials and Manufacturing team. So the vision of the Supply Resilience Forum is that the biopharmaceutical industry is considered one of the best understood and managed inbound supply chains of any industry in the world. To achieve this, the Forum leadership team set out 5 strategic objectives. These were: the inbound supply chain should be led by market insights, provide safety, resilience and quality, underpinned by end-to-end digitalization, open and transparent, responsible and sustainable. The webinar today supports the vision by contributing to objectives providing safety, resilience and quality, open and transparent, responsible and sustainable. The webinar is part of a programme of work that the team have undertaken. Initially, a paper was written with a lot of detailing called Navigating Cell and Gene Therapy Supply Chain Insights and Practices. There's a lot of detail in the paper, therefore the team have supplemented this with 3 webinars in order to give a high-level overview of the information and point individuals that are new to this area in the right direction. This is a live webinar, so you can post questions in the chat to any of our team and they can highlight within the document where you may be able to find the information. My colleague Ali has pasted the link for the document in the chat, and please just log on. You just got to put your name and your company in and it will allow you to download that document. It's a great piece of work. But now, without further ado, I'm going to hand over to Ike to introduce the webinar and start us off today.
Hello, I'm Isaac Young, a supply chain professional with over 43 years of experience in the biotechnology and medical device industries. I have experience in small biotech startups as well as large commercial firms, and have also worked on the supply chain for a supplier to the biotech industry. I have a multidisciplinary background in manufacturing, information systems, materials management, strategic sourcing, regulatory affairs, ethics, and operational resilience. I've been engaged with various biotechnology associations for about 20 years and have an MBA from Babson College. This webinar and the accompanying paper from which it is derived are crafted with the intent to provide some insight into the supply chain and related processes associated with bringing a new cell or gene therapy product into GMP production. It proposes disciplines that could begin being realized in early stage clinical activity that will help set the stage for a mature supply chain function that will support commercial operations. We will identify many practices that have been well established in biopharma and highlight distinctions of the more recent cell and gene therapy space. The intended audience for this work are professionals that have been involved in the disciplines other than supply chain who are now tasked with implementing the supply chain processes. Supply chain professionals that are new to cell and gene therapy may also find this work informative. As we go through the webinar, and as you'll see in the paper, we begin individual sections with a visual representation of the product development lifecycle. These chevrons have been color-coded to represent when we believe you should begin development of a particular discipline so that you can best optimize that particular discipline ahead of its absolute requirement and to avoid risk of cost or compliance issues later on. A green chevron suggests this is when you should begin working on that particular discipline. If you wait until the yellow chevron phase, then you're introducing more business risk at that point, such as higher costs or disruption to your timeline. If you wait until the red phase, you're introducing a potential compliance risk that may result in an audit observation or other issue that would impede your ability to bring your product to market. Please note, we would like to make this a living body of work that will improve with your feedback. Please contact BioPhorum with your comments, questions, and ideas that will help enhance this work.
Hello, my name is Amanda Sinquin. I am the Regulatory Affairs Manager at Fujifilm Irvine Scientific for our life science product lines. I have had a long career in basic research and academia before moving into industry to develop medical devices. That led to an interest in regulatory affairs and, in particular, the evolving landscape in the cell and gene therapy space, which is the next topic. The regulatory landscape around CGT products is evolving as the industry matures and the unique challenge it faces are addressed. However, this poses problems for suppliers and therapy developers to know what guidelines and regulations to follow. Even terms used in the industry either do not have a universally accepted definition or are not universally used. For example, the term ancillary material has been readily adopted as a subtype of raw material in some regions. However, the EMA in Europe does not use that term and refers to such materials purely as raw materials. In addition, the delineation between ancillary materials and starting materials is sometimes not completely clear. Another challenge facing the material suppliers and therapy developers is the lack of clear recommendations. For one, regarding the desired level of GMPs the ancillary material manufacturers should have to help the user reduce risk when performing qualifications. Another example is the importance of materials that are animal origin or animal component free. In this instance, there is neither an accepted definition nor a clear guidance as to the degree a material should be animal free in order to sufficiently reduce risk of adventitious agents. In general, ancillary materials are considered to be those that come into contact with the cell material during manufacturing but are not intended to be present in the final CGT product. An excipient is an ingredient that is intentionally added to the drug that is not an active substance. Starting materials are generally cells and tissues, but the scope can include all materials that will be part of the active substance. Labeling of ancillary materials is often research use only, which means that the products are exempt from most regulatory controls. The next level up would be for further manufacturing use. For both of these label claims, it is important for the user to further investigate what, if any, GMPs are followed by the material manufacturer and what release testing is performed. Some ancillary materials might be regulated products, either medical devices, such as some cell culture media, or drugs, such as certain recombinant proteins. However, even though this will provide assurance of the manufacturing site following GMPs and the product being consistent, the user still needs to confirm that the GMPs being followed and the release tests are appropriate for their intended use. Raw material grades can be compendial grade, following a monograph from one of the pharmacopoeia, for example USP grade. For other marketed grades of material, the user would need to ask for a definition. This is especially true of the commonly found GMP grade. Since there is no standard definition of what GMP grade means, this term can be misleading. Animal origin status of materials is important in terms of risk assessment for adventitious agent contamination. However, since there is no standard definition for animal-free status, it could refer to the ingredients, the manufacturing contact surfaces, the equipment used, or the facility. Once again, the onus is on the user to perform a thorough risk assessment and determine how much risk mitigation is required. There are several helpful guidance documents that discuss ancillary materials for cell and gene therapy, including USP 1043, ISO 2399-2022, and USP 92. These attempt to explain the various risks and considerations. USP 1043 provides a useful tiered approach for risk level of different types of ancillary materials and the steps that the user can put in place to mitigate them. The European Pharmacopoeia has a useful chapter on the risks of materials of biological origin, and ICH Q5A R2 includes a thorough description of strategies to reduce the risks of viral contamination from recombinant products derived from mammalian cells. Although not commonly found, some ancillary material cell culture media can be regulated, in the US as a Class II medical device for human ex vivo tissue and cell culture processing applications. Finally, the USP has several resources for particulates in injectable drug products. The take-home message is that a risk-based approach is recommended for evaluation of all ancillary materials. The responsibility is on the user to adequately qualify the raw material and to perform audits. Ancillary material manufacturers might be open to improving their quality management system and introducing GMPs appropriate for the therapy developer's intended use.
My name is Maya Sari Lim, and I am the Associate Director for Product Planning and Business Supply at Fujifilm Irvine Scientific. I'm a life science professional with diverse industry and academic research experience in cell culture media supply, stem cell bioprocessing, and bioprinting. Prior to joining the cell culture media industry, I was an academic at Nanyang Technological University in Singapore and then turned entrepreneur starting a bioprinting company. Today, what we see as one of the biggest challenges in cell and gene therapy supply chain is in finding dual sources for critical raw materials to minimize supply chain and shortage risk. And as part of our motivation in developing this webinar is really to be able to increase awareness and understanding of supply chain considerations for the cell and gene therapy related products, but also in hopes to gain better alignment across manufacturers and suppliers in defining raw material requirements. In today's topic, I would like to dive into supply chain considerations when it comes to the selection and definition of raw materials for the cell and gene therapy application. In regards to timing, as to factoring in supply chain considerations, we do highly recommend that this needs to start as early as the development phase. During this stage, it may seem obvious that the most important thing to do is to find a raw material that works. However, if we do not look holistically at the end-to-end process and identify potential quality or supply chain risks at this point, we may end up introducing undesired risks down the road. So, if we implement this later, such as in the preclinical stage, then we may introduce some business risks. And if we wait till phase 1 of the product life cycle, then you could potentially introduce compliance risks as well. The first step is to define the different types of raw materials. Here we have provided a table that summarizes categories of raw materials. First, we have starting materials that can include donor cells or tissues, cell lines or cell banks, vectors and viruses, or even plasmids, depending on the application. Some of these may or may not apply. Critical reagents for bioanalysis would include binding reagents, monoclonal and polyclonal antibodies. Excipients would include buffers, salts, polymers, and preservatives. Ancillary and raw materials includes cell culture media, growth factors, serum, buffers, and beads. Lab and manufacturing consumables could include things like filters, resins, tubings, thermoplastics, and electroporation consumables. Primary packaging would refer to single-use systems such as bioprocess containers or bags and cartridges. And last but not least, secondary packaging, which is often required for transport or shipping, and these include labels, softboxes, and cryoboxes. So what are some of the key considerations when it comes to the selection of raw materials? First and foremost, quality requirements is paramount in ensuring patient safety and efficacy of the final product. Raw materials used in the cell and gene therapy production must meet certain quality standards for the purpose of use. This involves defining critical quality attributes associated to safety, purity, identity, and potency. Another critical aspect related to safety and authenticity of the product is traceability. This is important to be able to track procurement and/or movement of raw materials throughout the supply chain to ensure this is met. When it comes to environmental health and safety, it is important to consider material or chemical handling requirements, facility requirements, and logistics or transportation. In addition, if you're planning to produce or distribute products to other countries, such as the European Union, there are also country-specific REACH regulations that one needs to monitor to stay compliant. For any cell and gene therapy products to be distributed for use, it is important to comply with regulatory guidelines and standards outlined by each country's regulatory agencies and division overseeing the licensing and approval. With respect to defining grade of cell and gene therapy raw materials, there's currently a BioPhorum team working on a comprehensive white paper addressing this topic. Thus, I will not go further into this here. The white paper is expected to become available in late 2023 or early 2024. When it comes to starting materials, whether it's autologous or allogeneic source, it is important to consider and understand that there are more stringent requirements, testing, and documentation when using allogeneic sources, such as detection of infectious or adventitious agents, as well as screening for potential immunoreactive markers. During the selection of raw materials, considering their availability with respect to supply chain risk and freedom to operate is also critical to minimize impact to production. Therefore, a careful selection of both the suppliers and the materials must be considered. Storage conditions and capacity needs for these storing raw materials to support production must both be considered to avoid any stoppage to production. Raw materials used to support production need to be stable throughout the production process and have reasonable amount of shelf life for storage to ensure product safety and quality. Last but not least, the cost of raw materials can also be an important consideration, impacting the final product cost, enabling patient access. Specific to supply chain, this table summarizes a few key points that one should pay attention to. Raw materials with short shelf life have a high inventory turnover, leading to high operational inefficiencies and likely higher cost. While products with longer shelf life is more ideal, one still needs to pay attention if quality can vary over time. Earlier, we briefly touched on storage conditions. Special attention should be paid to products that have specific temperature requirements, for example 2 to 8 or -20, and anything with sensitivity to light or even moisture. With respect to vendor stability, it is important to discuss production scale-up plans to ensure that the vendors are able to support increased volumes in batch sizes, process cycle time, receive and store materials in large volumes. Equally important with respect to handling large volumes is to consider ergonomic and handling requirements, and where possible, negotiate price and stability of price over time. Now, as you scale up production, it is important to continue to consider availability of raw materials at any period of time and possibly look at secondary suppliers that can support production volumes and meet timelines. In terms of logistics, certain raw materials may require temperature-controlled shipments, cold chain or ultra-cold chain logistics. When shipping across countries, packaging requirements may differ. Documentation requirements are also absolutely critical to avoid any delays due to customs. Now, the last thing you want to hear is that your expensive product is being held in customs for days or weeks due to inadequate or incomplete documentation. In the meantime, we still need to ensure that the products are maintained in adequate temperature requirements so as to not compromise the quality of the product. Finally, it is important to ensure that the receiving party has adequate equipment and facility to accept materials arriving. Sometimes it could be as simple as entry clearance or even the need for forklifts, etc. In addition, BioPhorum has previously published several articles in relation to raw material supply and approaches to risk assessment that you will find very helpful and relevant. So here we have highlighted 3 such articles that you can reference.
Now that we completed the selection process, the next step is to identify and generate critical raw material attributes. In the case of cell and gene therapy products, it is important not only to understand and define quality attributes of the final product, but also to understand and characterize each step of the manufacturing process. Since unlike biologics manufacturing processes, whereby purification and or filtration steps exist, cell and gene therapy products often do not have this step, and therefore quality requirements are often more stringent. Also, given the diversity of product applications, defining critical material attributes and methods of control specific to the manufacturing process for the product is therefore necessary. The most common and perhaps industry standard and systematic approach is to utilize the quality by design approach. Based on an article that BioPhorum has previously published, this diagram shows the 4-step process to identify critical material attributes. The first step is to define the target material profile. This is based on 4 key aspects: the intended use, quality criteria, safety criteria, and other requirements that impact product manufacture. The next step is to define the material attributes that are attributes impacting performance, so this includes the chemical, physical attribute, micro attribute, and other safety attributes. Specific to cell and gene therapy, special attention must also be paid to assessing risks of introducing particulates into the process from reagents, buffers, or single-use plastics or biocontainers, since most cell and gene therapy final products are intravenously administered. Step 3 is to review product control strategy. This involves a set of controls derived from current product and process understanding that ensures process performance and product quality. This would include defining process CQAs, critical quality attributes, and manufacturing process CPPs, critical process parameters. Establishing a control strategy can help to illustrate the impact of process step on downstream processes, product CQAs, and location of process controls, thus providing a holistic approach to product quality. In the final step, what we want to do is to try to bring everything together by defining critical material attributes and be able to score the impact by low, medium, or high impact. By applying this approach, both suppliers and manufacturers can greatly benefit in many ways, such as improving supply flexibility. By clearly defining critical material attributes needed to support this process, it can help both suppliers and manufacturers meet supply demands based on clearly defined criteria. Manufacturers also have a better understanding of the impact on changes in specification or supply chain. By demonstrating in-depth knowledge and understanding of the product and the process, suppliers and manufacturers can improve the quality of their regulatory submissions and strengthen the overall regulatory compliance and approval process. Implementing the BioPhorum quality by design approach can result in improved robustness of the product and the process, leading to enhanced efficiency, reliability, and overall performance. Finally, as materials are clearly defined and fit for purpose, there is less likelihood to over or under engineer the process and material grade requirements, thus reducing overall costs in sourcing such raw materials. In terms of risk assessment, we believe the ideal time to implement is at the preclinical stage. If we only start this process at phase 1, we may start introducing business and compliance risks by this time. So, what are some of the key considerations when it comes to risk assessment? First and foremost, raw material specifications play a critical role, as we have discussed already in the previous section, the role it plays in ensuring product quality and safety. Having a clear understanding of critical quality attributes to assess failure modes would be important to the success in cell and gene therapy product manufacturing. Performing risk assessment on supplier reliability is also important to ensure that your suppliers are able to deliver on time every time, but also provide competitive production lead times. Ensuring supplier stability from a financial, geographical, and political perspective is important to ensure business continuity. And finally, assessing supplier capacity, whether or not they have the ability to scale and deliver the same product quality at scale. By considering these key points and carrying out a robust risk assessment around each one, the team will then be able to gain good product knowledge that will enable them to build a strategy to ensure that they can achieve suitable process performance, state of control, and facilitate continuous improvement, while at the same time expanding the body of knowledge around their product and their process. We understand that it may not always be possible to find the perfect solution to address all of these challenges above, and some risks may not be adequately addressed by the suppliers. In these instances, steps should be taken to address risks associated by, for example, performing additional internal testing or increased inspections and process monitoring to identify any issues that may arise due to the variability of the product. In terms of how to perform risk assessment, fortunately, there are abundance of resources, and here are several resources that are already available and published by the BioPhorum group to provide guidance, tools, and tips on how to do this. So first, here we have an overview on quality management, optimal process, and RACI. The second reference here is to provide a holistic approach to raw material risk assessment through industry collaboration and supporting tools that can help one identify and prioritize challenges. And the last article here looks at both raw and starting material risk assessment for cell and gene therapy products and manufacturing. In addition, there are also USP standards that provide guidance on best practices and standard methods for cell and gene therapy. As an example, the USP 1043 is for ancillary materials for cell, gene, and tissue engineered products. We also have USP 1047 specifically for gene therapy products and so forth. Once the team has completed risk assessment, a final piece here is to make sure that we have a good communication plan, ensuring that both internal and external stakeholders, such as our supply chain team and suppliers, are involved, aligned, and aware of the risks. Working with these groups to ensure that robust plans are in place to mitigate risks is key, and ensuring that each group have resources to support such activities.
Consider the initial development of a sourcing strategy. Like much of other biopharma, the selection of suppliers and materials in the cell and gene therapy environment are often made at the bench in R&D, as opposed to through a classic procurement cycle. As such, the issues that might be involved with the use of new suppliers or unfamiliar materials are then inherited by supply chain to address and optimize for operations as you approach commercialization. With cell and gene therapy, there is likely to be a higher rate of materials being chosen that are new to the industry, some of which aren't yet suited to commercial GMP production. These will likely have a higher variability and less understanding of their critical quality attributes. Their initial specification is simply that they work in this new leading edge, little understood process, so just buy the same catalog number. The critical quality attributes that would be reflected in the true specifications haven't been developed yet. Some of these new materials may have legal or intellectual property concerns that need to be understood earlier on as well to ensure that they're going to be viable components of your process as you move through clinical development. Like the rest of biopharma, there's always a high cost of introducing change, whether you do it early on and perhaps take away from the resources that are trying to get a product to market, or at a later stage when you're faced with regulatory challenges of introducing that change. In either case, you're going to have a higher probability of ending up with a single or sole source critical material in your portfolio with evolving specifications to be secured. Getting at these issues earlier on and addressing their risk is a vital step to take. Engaging supply chain earlier on in the product development lifecycle will assist with the establishment of reliable sources by developing the relationship with suppliers so that they can understand and meet your requirements, or, if necessary, begin the process of identifying suitable alternatives for the materials and suppliers if there is not a reliable expectation of success with a particular source. A maturing supply chain function engaged early on can provide a number of benefits, such as addressing the legal issues and the contracting that will be necessary, managing supplier communications, establishing management disciplines, and even assuring the appropriate storage space for the materials is available. Supply chain can lead the development of total cost models so you'll better understand your future production costs. Supply chain will also establish global trade compliance and logistics functions so material can be moved compliantly across borders, whether it's international importing and exporting or even domestic material movement. In summary, the best sourcing strategy is to be involved earlier in the selection of the materials and suppliers, not as a gatekeeper, but to provide the business partner support to mitigate risk and optimize the evolution of a commercially viable production process for the new therapy.
Hi, I'm Leslie Varner. I'm currently a supply chain lead at Pfizer. I have 18 years of experience in the biopharma industry, with over 6 years being in material planning, sourcing, warehouse management, and logistics for clinical gene therapy projects. There are always challenges when planning, sourcing, or storing materials. These are three of the biggest challenges for CGT supply I have experienced. First, planning inventory of novel, custom critical, starting or raw materials with an initial short shelf life of 12 months or less due to ongoing stability studies. Second, sourcing these materials can be challenging due to the supplier's manufacturing capacity constraints, leading to long lead times, and alternative sourcing may not be cost-effective or a viable option due to project timelines. Third, CGT biological materials require ultra-cold or cryogenic temperatures, so assessing storage capacity, implementing cold chain management, and risk mitigation are essential to prevent material loss. To avoid project delays, it's best to start planning all materials, but especially novel, custom critical materials, as early as possible. Working with suppliers to understand their manufacturing ability and schedule material deliveries based on suppliers' capacity and project need, accounting for internal timelines and potential errors in supplier or internal manufacturing as well. My presentation will go into more detail about materials requirements planning, or MRP.
Hi, my name is Tom Walls. I'm an Associate Director of Supply Chain Planning at Spark Therapeutics, part of the Roche family. We have gene therapies in development and clinical stages and a commercial gene therapy product on the market. Keeping with our format of communicating optimal stage of development to implement these processes, we believe that these processes should be implemented during development. Having good MRP processes greatly reduces the risk of stocking out or not having enough materials to make batches per your project or operating plans. All industries which manufacture goods use some version of MRP in their toolbox to know what to purchase and when to purchase it. One quick note, during peer review of this content, a note was made that these techniques aren't specific to cell and gene therapies. That is true. These ideas and techniques have been around for decades. In the case of the materials requirement planning format we're going to review, or centuries in a few of the concepts we will explore. compared to the incredible science we are exploring and implementing to better patients' lives, these processes are very simple. But many cell and gene therapy companies consider these processes an afterthought and leave executing these processes to folks who don't understand the concepts discussed in this section. While the processes are simple, they require organization, focus, and discipline to keep the inputs required for these processes and calculations as exact as possible to ensure supply of the critical ingredients required to manufacture a batch. There are 3 information sources needed for materials requirement planning. First, the bill of materials. Know all the materials needed to manufacture a successful product batch. Maintain a list with essential information for sourcing the materials. Next, current inventory with expiry. Keep track of inventory amounts on hand and the associated inventory status. Lastly, the master production schedule or high-level project plan. If a detailed master production schedule is not available when material planning starts, a start date and planned number of batches can be used to get a jump on orders for longer lead time items and materials. Here's a look at the risks of not implementing MRP for any company. We can't understate the importance of supply of materials to support your manufacturer products. We all remember COVID and the state of supply chains during the pandemic. While we are thankfully out of that pandemic situation, the industry still grapples with very long and fluctuating lead times for many of the components we use. This was the situation before COVID-19. The pandemic exacerbated supply chain issues with existing material capacity issues. Those constraints largely still exist. But more than any other industry, our firms are much more sensitive to supply disruptions. The risks noted here are known to us all, but the role of inbound supply on these risks is often not understood. Having a disciplined MRP process and trained professionals who know these techniques is an important risk mitigation for any cell or gene therapy company. I'm going to hand it off to my colleague Leslie, who will look at some of these processes and techniques in a more detailed light.
Thank you, Tom. We will explore MRP components, key relationships, and benefits in greater detail at our second webinar. We will also review the MRP template tool for new startup businesses with development teams or individuals new to supply chain principles and practices in our third webinar. Registration is free and open for both webinars now. Please use the QR code or click the link on the next 2 slides to register for each webinar. Hope you can join us.
So we'll add the QR codes now. Ali will show those QR codes while we open up the chat and ask for any questions for Tom or Leslie on MRP. There will be a more intense deep dive into these subjects in our second webinar. So the 2 webinar links are in the chat. And so in a moment or 2, we're going to move on to supplier selection and supplier qualification, which Donnie and Ike will take us through. So there's the 2 QR codes. You can scan the QR codes using your mobile phone or use the link in the chat. As Leslie said, she'll be deep diving into these subjects in a little bit more details. So we're looking at material qualification and qualifying the supplier, and Donnie and Ike are going to take us through this one.
Hello, my name is Donnie Beers. I am the applications leader for cell and gene therapy at Integris Life Sciences. I have been in the bioprocessing industry for about 15 years in capital equipment sales, single-use technology for both upstream and downstream processing. Today I'll be talking to you about material qualification and qualifying the supplier.
When is the best time to conduct material qualification? In our view, the ideal time to do this is as early as possible. Preclinical phase is the ideal time to implement. By the time you reach phase 1 clinical trials, at this point you are starting to introduce a business risk by not implementing. As you progress towards phase 2, you may also be introducing compliance risk by not having qualified the raw materials or materials at this time.
There are a variety of reasons why you may need to qualify a new material. It may be due to getting a new grade of an existing raw material. It may be due to getting a new-to-world technology that's superior to others that have been used. It may also be due to a corrective or preventative action. It also could be as a result of wanting a second source for security of supply, and you're simply qualifying another similar material. Regardless, you need to start at the raw material specification. So to this point, if you've done well setting up your raw material specification, it flows through a lot easier for the qualification plan and how you need to bring this material in. If you have not done that at this point, this is the time where you need to meet with the supplier and confirm agreement on the specifications. At that point, you'll be able to do a gap assessment where both parties can agree who and how any gaps will be resolved. As always, it's best to take a risk-based approach, identify and document the control or qualification points that you're going to need. And then from there, you can define your control and qualification strategy. Starting with the highest risk, all the critical quality attributes will need a plan and an owner so that they can be either qualified or controlled. This also gives you an opportunity to have a mutual schedule regarding deliveries of material and information or reports that will be exchanged. And it's important to communicate and update the schedule frequently. And then at that point, you're ready to execute the qualification and control strategy. It's important to note at this point that you may want to review any emerging guidance or regulatory concerns and compare them to your current plan. Now, as for where there are some critical areas in supply chain to consider in your efforts of material qualification, one of the easiest ways to get derailed is to have an expectation that you are going to get multiple lots of material very early in the process. And if you simply place an order, the vendor may not know to produce that in 3 separate lots. So communicating this early allows them to have time to accommodate that into production schedules, to give an accurate and realistic lead time, and make sure that there's no miscommunication. Another one that can be a stumbling block is discussing payment terms early. If you have some flexibility in the ordering process on both sides, this can help it go a bit smoother. But when you're getting small orders of new raw materials that haven't been qualified yet, most payment terms are set up for large volumes of business. And so you may need to consider alternate strategies for how you can get that material in, either through a distributor or something else to make sure until you can get your supply contract agreed to, you may need an alternate method if the payment terms don't line up. Throughout the whole thing, it's important to communicate the total forecast for development qualification materials. This is critical so both parties know the expectations and when critical dates are. It also may come down to a little bit about the relationship between the end user and the vendor. Some companies are naturally more collaborative and some are more transactional. And considering that into your plan is going to be important. Another thing that can trip you up is if there's multiple sites involved. Getting those sites set up in different purchasing systems and making sure that the vendor knows that those materials can go to other sites. This will save you time down the road if you get those sites set up early. Some companies also offer verification and validation services. This may be something that can help you get to market faster. Around quality and supply agreements, it is our view that you should establish these as early as viable. This also would include things such as if there's anything that goes wrong, a shipment is damaged or something, a timing on replenishment of inventory due to unforeseen circumstances should be considered. Another thing is around the timing of product ascension to the final specification. New developmental materials, it may take a period of time before they are ready for GMP use, and making sure that both end users and suppliers' timelines are lining up. One thing that should be considered in your risk assessment is if there's any considerations for hazardous material logistics. So the material needs to be safely taken from one spot to another, and make sure you incorporate that in your plan. One other thing, when you have highly variable raw materials, make sure you dedicate time if you want to receive materials on the high end and the low end of that, to make sure it doesn't have any impact on your process. And that should be communicated early, because those may need to be engineering batches that would not fit normal production schedules. And then, of course, as with anything in the emerging field of cell and gene therapy, it's important to keep abreast of changes in guidance and regulations.
When is the best time to qualify the supplier for critical materials in your cell and gene therapy process? In practicality, it is best to qualify the supplier as soon as you are able. However, to give yourself the most time in order to make sure everything works out correctly, the ideal time to implement may be when you do your BLA submission. The reason for this is you know your process, everything is locked in at this point, and there's probably going to be no further changes. However, if you can do it sooner, that is a good thing to do. But if you wait until post-marketing, you are introducing business risk by not implementing this. While it is not a requirement, as long as you are following your quality management process, it is a risky proposal to do so.
So what is the important part about supplier qualification? Especially in late phase and clinical and commercial, the supplier must maintain control of the goods and services from their upstream suppliers. And so end users are responsible for periodically assessing the quality system, including CAPA controls, to make sure that is handled well. So as we use a risk-based approach, the risk tier will dictate the depth and frequency of the assessment. If it is a critical raw material, maybe has some variability to it, and it's a sole source, that may end up in the highest risk tier and should have more frequent monitoring. Anything that is deficient in the quality system or in the products or services will need more frequent assessments. However, findings are not necessarily always bad. So responses to findings and the absence of findings are a litmus test. So this is a two-way street. It allows you to establish a working relationship and expectations. It is our suggestion that every organization try to better themselves by taking in that feedback. All feedback is good feedback. So suppliers that welcome that guidance to aid in their continuous improvements are seen as better than some who resist undertaking remedial efforts, especially where they're necessary in these critical raw materials. Another thing that can be used is external services, such as contract auditors, can help supplement, add efficiency, and increase standardization in assessments. So in establishing this relationship, you can make use of other good tools out there in the market to gain quick information on the quality of your supplier, but this is not a substitute for on-site audits. It is an addition that you can use, but it is not a substitute for the collaborative dialogue that can come out of this, where both parties can end up in a better situation. So even some findings that on the surface may seem difficult to resolve or expensive to remediate can ultimately put both parties in a better position if they can collaboratively work out how those issues get resolved.
Change notifications. We mentioned this earlier when we discussed quality agreements, but again, you need not wait for a quality agreement to establish this important function. Some suppliers already have a fairly elaborate mechanism in place because, again, they know this is an expectation that will be coming from all of their customers. If so, it may be as simple as identifying which items you want change notification on. Do inquire of the supplier's willingness to provide those change notifications in advance of any change, so you will have more time to assess and adapt to the change without disruption. The other half of establishing this mechanism is developing the internal process for logging, evaluating, adapting, and documenting the change within your processes and systems. There is an initial triage that is required to weed out the inconsequential notices from the serious ones and the urgent ones from those that can be accommodated with your standard change control process. Identifying the subject matter experts that will be needed to review incoming change notifications and ensure that the evaluation is done in a timely manner. You could also consider putting this on your supplier scorecard to further refine what you consider to be important notifications and how timely they were delivered. Material issues. Being in the earlier stages of the learning curve associated with developing technologies, you're likely to have more material issues than in a more mature manufacturing process. Many of these issues may not be supplier related. There may be an absence of understanding of inherent variability in the material, or perhaps the actual specification requirements have not yet been determined. One of the key diplomatic roles that you'll be playing in supply chain is how to manage that, because quite often the finger gets pointed at the supplier too soon. If it does turn out to be a quality issue with the supplier, a formal means of notifying the supplier of the observation and expectations for corrective action should be employed. The supplier corrective action report is a common tool for identifying the issue and the timeline necessary for an investigation and corrective response. Internally, the deviation tracking and change control processes should be robust enough to document the SCAR and the supplier's responses. They are also a key topic at the business review with your supplier. Risk management. A business continuity planning program isn't likely to be happening at an early stage company. It's generally difficult defining protectable assets at this point. There's certainly shareholder value to be protected, but you're already in a high risk tolerant stage of a risk tolerant industry, so you're not likely to find the board of directors asking for these plans. But that said, there are things you can start doing now that serve multiple purposes. If you build risk awareness now and you start logging and tracking that risk, you create that internal awareness so that things aren't proceeding without taking the time to stop and ask, how reliable is the source of this material or service? Is it coming out of a challenged region of the planet? Is it derived from something that's going to be gone later on? Is it something that's going to be subject to future regulatory controls that may limit its availability? Logging and tracking risk does not mean you need to be reporting it necessarily or specifying immediate remedies. Certainly, if you have something that's egregious enough to warrant prompt action, it should become a speaking point internally as well as with your supplier. But discussing risk in your business reviews will convey to your suppliers that this is an important topic and that you're going to be increasingly inquisitive about where they are in their efforts around resilience. There are a number of suppliers in the biopharma space that have well-established programs. They are leadership models with regard to risk management and business continuity and can be a great resource in this area. Also, benchmark with your peers. BioPhorum and other associations have groups dedicated to this. BioPhorum has been working on publishing a number of important documents on the topic that can help guide you on your individual efforts. Most companies don't compete in this area, so it's a good idea to share best practices and documents. Transitioning suppliers. As you get into the later stages of the product lifecycle and you're getting close to commercialization, compliance with all aspects of quality systems becomes mandatory. If you've started early and you've been working on your supplier relationships, then you'll understand which suppliers are ready to go the distance with you and which ones need more attention. This is why it's better to start early in this effort. Elements such as quality agreements, change control, performance measurements, supplier audits. These will become documented parts of your quality systems and are reviewable by regulatory authorities. Focusing early on will also assure that there has been sufficient dialogue on forecasts and your supplier's abilities to support your requirements. The inclusion of your critical suppliers in the demand and operations planning process will yield a stronger, more agile plan that is supported by nurtured dialogue and aligned expectations. Last, let's talk about how, as an organization, you implement some of the things we've been talking about. A RACI chart is a good tool for looking at this. For those who don't know, RACI stands for Responsible, Accountable, Consulted, and Informed. And it's a means by which you can document what role individual groups or people are expected to play in some of these elements. This is an example chart, don't use this one, but it's an example of where individual groups would be expected to factor into the realization of selected elements. This is about looking at how supply chain is building the connections in the organization to support the delivery of products. Who wants to be informed, who must be consulted, and who is acting on it. It becomes a means by which organization understands all the resources that are involved and all the fingers that touch what's going on. The creation of a RACI is more than just generating the document. The more important takeaway is to go through the exercise of considering what groups are or should be involved and how they're involved and what does that involvement require. Project teams and plans will then evolve from this along with the quality system documentation of your operations. Supplier qualification and relationship management is a broad subject area, and so rather than trying to cover all the additional elements in this webinar, the team has produced a follow-on second webinar which explores in more detail some of the other things you'll want to be thinking about, things like scorecards and supplier segmentation, so you can balance your resources appropriately. Business reviews, quality agreements, master supply agreements. So I would encourage you to check the link in the chat and or get in touch with BioPhorum to sign up for the next webinar, and we'll dive into these topics a little bit more at that time.
Thank you, Ike and Donnie. I appreciate we're having a few technical issues on the call today. Donnie, would you like to add anything to your section just to clarify any key points?
Yeah, thanks, Vicky. So one of the things that was discussed in the material and supplier qualification section was the times when you should do certain actions. And so when should you start supplier and material qualification? You should start those as early as possible. Phase 1 and phase 2 is when this activity should begin, and that will be required for IND submissions and things of that nature anyway. So the thing that's different about cell and gene therapy is that the material and the supplier may not be mature enough yet at the point when you start that journey where there's a research use only material, or it's produced on an engineering batch basis, and it's not the final manufacturing process, maybe not the final manufacturing location. And so before you sink all the effort into validating the raw material and getting your supply and quality agreements in place, it's important to make phase appropriate choices. So begin that activity early and do an appropriate level of qualification at that time. And then when you get to your BLA submission, that's when you really, that late phase 3, as you're scaling up, you're starting to buy commercial quantities of materials, that's when it's worth spending the effort and the money and doing that full validation and qualification of your materials and your suppliers. So we did a really good job of discussing that in the paper, so I would recommend you check that out. It's nice because you can just zero in if there's a particular section of interest, just go read that one section. It also references other BioPhorum works. So it's an excellent way to kind of increase your knowledge base with some targeted reading. So thanks, Vicky.
Thanks, Danny. And I'm just going to put a copy of the document in the chat. And as Donnie said, it's I think Ali's beat me to it. It's a really great document. We've tried to give some highlights today, some thoughts, some just a high level overview. Within the document, we deep dive into each of those sections, and within the following 2 webinars, which we're going to put on the screen again in a moment, we really deep dive into that MRP. What's important from a cell and gene therapy? Are you from a research background, or are you a bench chemist that inadvertently may be locking in supplier decisions now during your experiments without potentially understanding the full impacts? By working closely with your supply chain, this document really highlights the opportunities to really accelerate the commercialization of your product and not fall into many of those traps that I'm sure there's a couple of people on this call have fallen into when developing and taking a product through to commercialization. Okay, so if there's no questions for our audience today, I will bring the webinar to a close a little bit earlier. I'll thank everybody for joining us. I apologise about the technical issues we've had today. We certainly will be looking to iron those out. We will keep clean up the presentation. We will make it available for anybody that would like to share it internally within their organisations. As you can see, it's really great for those new businesses and those who are new to the cell and gene therapy area. It really highlights and it brings out some of the things to look at just in case, so you don't fall into those traps. And I'd just like to thank today Donnie, Amanda, Maya, Ike, Tom and Leslie. I know you've put a lot of work into this webinar. Thank you all very much. It's been a pleasure working with you on this.